Okay, so this is about classifying organisms and where exactly, or how you would like, it's making a diagram where it's like a visual classification. This week's for you guys, you'll have an ed puzzle that is about taxonomy and like old-fashioned classification where you put things into like kingdoms and phylums and class and order and all that stuff. So that's fine because that's like creating a name for an organism. But this is like a system where it's like a visual, like how closely are these organisms related? And that's basically what we're going to be talking about today. And it shouldn't take too long. I'm going to take you through a few vocabulary points. And then we'll go through some examples and we'll practice reading them, which is the important part. Like usually if we were in class 5 days a week, we'll like make them. Like we'll like have some organisms. Like I even have a handful of like candy here today because I was like, well, if I have time, like I have all these different kinds of candy and we'll pretend like they're organisms and what do they have? Like what are their characteristics and how can we put them on a cladogram? But I don't know that we'll do that today. But anyway, I have it just in case. But that's what we would do in class. However, your job isn't, as a biology student, your job is not to create cladograms. Your job is to read them. And so you've already done some cladogram creating when you were doing your NOVA deep trees, although we call those phylogenetic trees because they're shaped more like a tree. And this is called a cladogram. And unfortunately, I don't think I wrote that anywhere. Cladogram, so C-L-A-D, and then it'll be O. See, there's clade over here. So it'd be C-L-A-D-O gram, G-R-A-M, cladogram, okay? So a cladogram is basically a tool to kind of visualize relationships in between organisms. So you can see like how that, almost like a family tree, like how they're all related to each other. So what you did on NOVA Deep Trees last week was you did like phylogenetic trees, and phylogeny just means like classification. It's kind of another word for classification. But maybe you can see a difference. Like the ones you did rooted at the bottom, so their root was at the bottom, and they kind of just branched out. And it was just kind of like, oh, if it goes this way, you know, if it's got whatever characteristic and all that. This one is left to right typically. Now you can have them twisted so that their branches like, you know, up and down. So you can have that happen. But this one is more typical of a cladogram, is left to right. Okay, the thing about cladograms that are different from phylogenetic trees, which you did on that little activity I gave you at the end of last week, is that cladograms kind of indicate time. So you could actually make a graph. Like if you wanted to, you could like put an axis here, and you could put an axis at the bottom, and you could put time on this bottom axis. Because cladograms will indicate time, where phylogenetic trees, what you made on that little activity, don't have any like time involved at all. It's just how closely are they related. So this could be like, you know, I don't know, 500 million years ago or whatever. Okay, so because of that, because time is involved in this, then this would be an ancient like ancestry thing. Sorry, my phone's blowing up. This would be like an ancient ancestor that would be extinct, right? Whenever the timeline stops for an organism, like you can see right there, it stopped for this organism. Then that means that that organism is just a common ancestor and it's not here anymore, okay? So who is this? Like if I have species A, B, C, like who is this? Okay, I'm not gonna lie to you, most we don't know. It's just we know that they've branched off of some common ancestor and we don't know what that was. Now, sometimes they can sort of find a common ancestor between two species, but it's pretty rare because it's usually way back in the fossil record and you have to like really work between, you know, trying to figure out, okay, was there another intermediate species or was this the one? And that's kind of hard to do. But we do indicate that this was a common ancestor between these and it stopped. Its existence of that species stopped, okay? So what we have here then is a branch point, right? So it's branched off and we have another branch point. And so these two that are right here together on this clade together, we call those sister species, okay? Because they are very closely related. And so those would be our sister species and they have, okay, there's our common ancestor of all of them. Kind of maybe put these in a weird order. Think about that. So this little branch back here is going to be the common ancestor for A, B, and C, all three of those species. And then, of course, we have a little branch point here. And then, oh, a divergence again. So they branched again. Okay. And so there was a common ancestor of A and B right there. Okay. So, excuse me. I'll give you some time to write that down. While I choke on my own spit. Okay. So basically what you're seeing is this has all to do with speciation. So imagine, last week we talked about all of the different types of speciation, of a selection, like directional selection and stabilizing selection and all that stuff. So imagine you have organisms that are in two different areas that are going through different types of selection. Like in one area, they're having a directional selection one way, like that particular environment is best for that extreme. In another area, other extreme. After a while, they can't even be in the same location together because mutations have built up so much in their populations that their DNA isn't even close enough for them to like mate and produce fertile offspring, which is what we have to have a species. So that was them right here. And so then what happened is it diverged and we had a divergent evolution occur. So they diverged from each other. They were differences. And we ended up with a C species here. And then we ended up with some unknown common ancestor of A and B. Okay, those were the two that diverged there. Time went on. And apparently the environments were different for A and B. And they had a speciation event where now we have two different species there. Okay, so those are now sister species. They're very close to each other. They're both related to C, just not nearly as close as they are to each other. Okay, I need you to know node, that is called a node where you have a branch point called a node. I need you to know that this is an outgroup because it's the least related to all the rest. So basically it diverged and all by itself. Okay, so I need you to know the outgroup term. I need you to know node. I need you to know how to find a common ancestor of either a sister species, which would be at that node, or a common ancestor of all of the species would be at this node, or this little root, if you want to put it there, but anyway. If these are sister species, and a little box-like thing is called a clade. Now, this would be one clade, but guys, you can back up and say this thing is a clade too. It is. And sometimes you can have 10 or 12 clades on one cladogram. Okay. Questions about the vocab and what this thing is? Any questions so far? We're going to look at some. Okay. Bumping right along. All right. So these clades right here, these are two different ways to draw a cladogram. So this one's more like a little tree where you have the root down here. You're still seeing an outgroup, right? See this outgroup right here? You're still seeing an outgroup just like you are here. Okay, and this is a cladogram that's turned up on its end. You have to kind of just be okay with them being in different shapes and in different orientations. Like you never know what way you're going to get it. So you kind of have to get used to reading it. So here's your common ancestor of all. Here's a branch point. C is the outgroup all by itself. B and A are sister species. Okay, and their common ancestors right here. Over here, let's look at this one. Here is your common ancestor of all, right down here. Here's your outgroup. It branched off by itself. And then A and B are on this same node together. So they're sister species. Okay? Questions? Sorry, I peppermint my mouth. I was coughing. Okay, so now here is a rule that is super important. We say that species can rotate freely on the node. That means that on this node right here, this little point, A and B could just flip-flop. They'd be equally related to C, okay? They can just flip. So you could just as easily have put species A here and species B here. Okay, does that make sense? They're equally related to C. Just because A is in the middle doesn't mean it's closer to C than B is. B is just as equally related to C as A is. Same thing here. A, B, C. I can flip-flop around this node. So I can just go whoop and flip it, and it could say BAC. Exact same. All right, cool. All right, see some nodding, good. Okay, sometimes you will see cladograms that have like morphological things on them. Now, when I talk about morphology, like MORPH, morphology, I'm talking about things that I can like either see on the outside or that I know exist as body structures on the inside. I'm not talking about DNA. Morphology is like structural things, adaptations. Okay, so like this, hair and fur. Well, that's morphology. Okay, so sometimes you'll see little traits like this on it, on a cladogram, and you're like, I mean, what's the purpose of this? So I can ask you questions about this because we're looking from left to right on this particular one, but sometimes it'd be right to left. But basically, it's like a graph. As it goes up, that's the way you're reading this. Okay, so let me back up. As it goes up, that's the way you're reading this. Okay, so you see what I mean by that? So as it increases, no matter if it's left or right, then that's the way you'd be reading this, because this little red line that says vertebrae is telling me that everything past that has vertebrae, has backbone. Okay, so everything past that line has a backbone. Cool. This is 4 limbs right here. So that means that everything past that has 4 limbs, but this one doesn't. So obviously fish don't have 4 limbs. They have fins, but they don't have limbs. Okay, so now everything past this should have 4 limbs, including birds, which have wings. Those are still limbs. Okay, all right, so amniotic egg. Amniotic egg is falling right here. You may not even know what that is. But if I said, which of the following does not have an amniotic egg? Then you just have to look at it and go, amniotic egg is right there. Everything past it has an amniotic egg. Anything before does not. So frog does not, fish do not. Cool. What things have eggshells? What's going on with the eggshells?
Would that be just lizards and birds?
Just lizards and birds. It's absolutely correct, because it's put on their branch and not on the rest of them. And it's done that way because this is an adaptation that they have that just seems to have arisen after they speciated from the main group. Okay, now they are on a node together, lizards and birds. Kind of funny to think about, but that is what scientists believe, that lizards and birds are actually very closely related to each other. Then we have hair and fur with our rodents and gorillas, and our rodents and gorillas are both mammals, so they are actually sharing a node. Okay, good. Got all that internalized? Everybody good with that? Okay. Oh, fun. This has numbers. It's basically a percentage of divergence, like how different they are from each other, but just ignore the numbers for now. We're just going to look at the names of the bears. Okay, somebody tell me which species is most closely related to the sun bear?
The sloth bear.
The sloth bear. How come it's not the Asiatic black bear?
It's not a sister species.
Right. Even though they do share a common ancestor right here, the sun bear and the sloth bear, which now you can't see because of my toolbar, share a more recent common ancestor, right? So it was more recently where did they diverge from each other. Okay. Let's see. There's nothing else exciting about this. Polar bear and brown bear, sister species. They're both closely related to the American black bear, but they're more closely related to each other, polar and brown. So brown is a grizzly bear, by the way. Okay. Is everyone able to read this okay? Once you figure out how to read them, they're pretty easy to read, honestly. My AP kids love it when they put cladograms on the AP test. They're like, I got that in the bag. Okay, so all of this, well, not that one probably, but all of these and before, we were kind of basing on morphology. Like, what does it look like? Or what does it have? Like, what structures does it have? Cladograms now are being based on molecular evidence. Remember, molecular evidence is kind of our biggest evidence of evolution because we can look at actual genes and see what the heck is going on. So I have, I'm going to switch my face camera over to my document camera. that you can see what I'm looking at in front of me as well as myself at the same time. There we go. Only now I can't see it. Let's turn some light on that thing. Okay. So let me see what you're looking at. Cool. All right, so this is a little chart that we can use. That basically what you do is you count differences in these things. So basically horse versus human. Now what you're looking at, let me go back to I got so much stuff going on here, I don't even know where I'm at. Here I am. What you're looking at on this particular presentation screen is you are looking at the number of like amino acid sequences. So that would be like protein, you know, like proteins are made out of amino acids. And so, you know, we use that little amino acid, that codon chart to figure out like what our DNA coded for, like what our mRNA coded for. And we were like methionine, glutamine, glycine, all that stuff. So that's kind of what we're looking at here is the actual like thing that was on that codon chart on the inside. So that's glycine, proline, threonine. I don't know what that one is. Anyway, okay, so in red, you have the human. This is for cytochrome C, and cytochrome C is one of the proteins that we use to in cell respiration. And so it's like really highly conserved is what we call that. That means that over evolutionary time, that protein has been conserved very well. Like almost all organisms have cytochrome C, even like plants and stuff. And so this is a good one to use for cladograms because you have a lot of organisms that have it. And so you'll see a lot of these with cytochrome C if you stick with biology. And so this is basically saying in point 1, so the first amino acid in this particular section of cytochrome C for humans is this GLN, right? And then it's proline, and then it's tyrosine, and I can't remember them all. Serine, threonine, alanine, lysine. I'm doing pretty good. Anyway, so that's our order, right? So what we're trying to do on our little chart here, and I put human versus human, there should be no differences between them, right? Can you guys see my little chart thing? Oh gosh, now you guys all disappeared on my screen. Whatever. Okay. My Chromebook just lost its network. It does that all the time. Okay, so basically what you're seeing here is, oh wait, tell me one more time because now I'm on here. Can you guys see this little chart? Like where my face is supposed to be? Okay, I don't know if it's like readable or whatever. All right, so what you're supposed to do is compare them. You're supposed to count how many differences are between the human and the horse, say. Okay, so here's the horse up here. So I'm going to look at the red versus horse. I'm going to say not different, not different. Oh, there's 1, 2, 3, 4, 5. 5. So then I'd write for horse versus human, I would write 5. Let me get my pen. Okay. I would get 5 for horse versus human, and there's 2 of them. Human, horse, horse, human. So I do that twice. Okay. All right. And then I would do human versus rabbit. Okay. So then I count those up, and I'd be like 1, 2. Oh, I'm down here, by the way. 3, 4. 4. Rabbit versus human. Okay, so now I've got a job for you. I'm going to see who all I have here in my little Google Meet. Just in case. If somebody else has got a different number, let me know. Okay, let's see. So now what do we do with these data? Like, what am I supposed to do with this? Let me switch over to here. What I have now is my little chart that I made, okay? And now what we would do is we would attempt, now notice this is differences. So that means the smaller the number is, the more closely related they are. So we would attempt to create a cladogram. Now I'm going to tell you right now, not easy. Okay, drawing your own cladogram is actually really hard. So you'd be like, all right, I'm going to start over here, okay? And I'm going to make some branch points. So I got to kind of figure out what's most closely related to what. And yeah, no, you can't even see that. So we'll knock some stuff off and then we'll move it up. All right, so looks like we have some, sorry, it's being naughty. Some ones right here. Look at this one. super closely related. Human and a monkey. Okay. Also, the next one is going to be like this rabbit to monkey, and then oddly enough, rabbit to chicken. Like, I don't know what's going on there, because I would think that we'd be more closely related to the horse than the chicken, but you know, whatever. Sometimes you get surprised, okay? So basically what you'd end up doing is you draw little clades and you would say, okay, human and monkey, because they're close together, okay? And then the furthest from the human is going to be horse. Something wrong with the horse's cytochrome C. I don't know why that mammal. Horse is a mammal, so it shouldn't be so far out. Monkey for chicken. So that totally makes sense to me. Like the chicken and the, what's that chicken? Like, look at all these high numbers for the chicken except for to rabbit, which I'm wondering if we counted that one right, but whatever. So those are all kind of high numbers comparatively. And so typically you'd like put the chicken out here. Now we have a little problem because of this one right here, but whatever. So then you just kind of try to figure out how your cladogram should work, okay? And then you'd put more clades on as you went along, okay? So this would be your out group right here. And then these would be sister species, okay? And then I probably should have branched a few more, but this is going to be ugly. But then you just put like another branch point here, and you more than likely you should have your two mammals on here, but it's okay. So you'd have like your rabbit here, and then we have an issue with our horse. I don't know what's wrong with our horse. It should be up here with them, but seems to be on its own little line. So what you could do, you could be like, this is going to be the horse, okay, and just ignore that. So anyway, so this is how you would like try to create your own cladogram, right? So you're just creating, I'm going to fix my rabbit line. So you're just creating it based on molecular evidence. So it's a lot different than saying, oh gosh, well, the human and the monkey and the rabbit and the horse all have hair, right? Or fur of some sort. So we need, this is like, oh no, in their protein, their cytochrome C protein, this is how many differences they had. So of course you may have a slightly different cladogram if you used a different protein, like if you were looking at a different protein structure. Or you could look their entire DNA sequence and be like, how many differences are in their DNA sequences? You could do that too, okay? I mean, obviously this is rough. It's like, I mean, you probably want to spend more time on it, but that's basically how you do that, okay? Questions? Questions on cladograms at all? Does that make total sense? Okay, so that was our 30 minutes of class, and then I have about a 15-minute little worksheet for you where you just go in with your notes that you just took, and you just say, identify the sister species, identify the outgroup, and then look at a cladogram and try to tell what's most closely related. It shouldn't take you more than like 10, 15 minutes, okay?